Alright, so now we're going to get down to oxidative phosphorylation. This is where everything that we've been going through to generate NADH is really going to start to pay off. So again, following a molecule of glucose through cellular respiration, we started with glycolysis, which basically got us some NADH and some ATP and 2 molecules of pyruvate, to the citric acid cycle, where we oxidized that carbon molecule completely, generated more NADH, a little ATP, some FADH. And so now we're going to focus on all those electron carriers. All the NADHs and the FADH2s that we've got, we're going to feed them into what's called oxidative phosphorylation, or the electron transport chain. All right, so this is the purple box here. This is the last major step to generate lots and lots of ATP. So ultimately, this electron transport chain is just what it sounds like. It's going to be centered on the inner membrane of the mitochondria. Again, what do proteins do? They do stuff. In this case, we've got a bunch of proteins in that membrane that carry electrons. And they carry electrons in a way that they take these high-energy electrons and kind of hand them off to the next protein, to the next protein, to the next protein, each at a lower level. And that's how we kind of draw energy stepwise out of these electrons. So you can see the major complexes here, complex 1, 2, 3, 4, and then finally ATP synthase, all involved in ultimately making ATP through chemiosmosis and oxidative phosphorylation. So the electron transport chain itself is going to be complex 1 and 2 feeding into quinone, complex 3 feeding into cytochrome C, and then finally complex 4. All right. So what is the point of this? Again, is we're taking the energy from your food, and ultimately we're going to make a proton gradient, and we're going to harness that proton gradient to make ATP. So let's look at this. So again, here are our NADH and our FADH feeding into the electron transport chain proteins. And as I hand off through the various proteins in those complexes, the electron, I can use it to pump protons against their concentration gradient. That's what the energy from your food is used for, to pump protons against their concentration gradient. All right? So again, we're taking the high energy electrons from NADH and FADH, passing them through the proteins. Cytochrome is a term that you're going to hear again and again as you go on in biology. And each of these has a metal center, and that is important in the redox chemistry. So cytochromes have this metal ion bound to them. The electron transport chain itself doesn't generate ATP, right? The point is to pump protons across the membrane. Now, oxygen comes into play here because it is our terminal electron acceptor. That terminal electron acceptor is the lowest energy, right? So we go from our highest free energy to our lowest free energy, and hence the difference is energy that's potentially usable, in this case to pump protons. Right? So down at the end, you can see of complex 4, there's 2 protons plus oxygen yield H2O. So we've taken that and basically oxidized those, in essence, those electrons as best we can. We've taken the energy out of them so that oxygen is the only one that still can be reduced because it wants those electrons so badly. It's so electronegative. Now, what are we doing with all those protons? Is we're building up a gradient, right? So if you look here, see how we're pumping the big arrows push up, 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 up through complex 1, 3, and 4? Those are our proton pumps. The protons then accumulate, in this case, in the inner membrane space between the outer membrane of the mitochondria and the inner membrane of the mitochondria. And if you give them a channel by which they can flow down their concentration gradient, in this case, that channel is the ATP synthase, you can use the flow of those protons to literally move to power this wheel that is the ATP synthase, and that, as it turns, will literally make ADP plus inorganic phosphate into ATP. Okay? So a very, very important enzyme complex, the ATP synthase. So this is what we call chemiosmosis. So we're coupling the flow of electrons down there, the flow of protons down their concentration gradient across the membrane through the ATP synthase to make ATP. Second law of thermodynamics. things going from regions of high concentration to regions of low concentration, right? From regions of high stability, or sorry, high order to low order. I'm headed towards equilibrium. So we call this chemiosmosis, the use of that proton gradient to drive cellular work. So again, this is the proton motive force, as we've talked about before when we talked about the use of cotransporters, the buildup of the membrane potential. It's the driving force behind ATP synthase. So again, the PMF has two components: is the chemical component and the electrical component. So the delta pH and the delta psi. So those two together, only summed, do you get the total PMF, and that is the force that we tap to make ATP. Right? So during cellular respiration, most energy flows from glucose to NADH to electron transport, generate a proton motive force to make ATP. About 40% of the energy in the glucose molecule is transferred to ATP. We make 38 ATP total. So now take a look at this summation. So glycolysis, going from glucose to pyruvate, we get 2 ATP. In the citric acid cycle, we get 2 ATP. Now, all the rest of the energy that we can harvest from glucose now is in the form of these high-energy electron carriers that we feed into oxidative phosphorylation, the electron transport chain, to build that PMF up. And now, from the chemiosmotic mechanism, we get 32 or 34 ATP. It's hard to quantify that exactly. So you can see that this is very, very efficient as opposed to substrate-level phosphorylation. Right? So about 36 or 38 ATP is the number of ATPs that we're generating from a single glucose molecule. Right? Now, just to mention it, we look at this process in aerobic cells. So cellular respiration in aerobic cells. Aerobic meaning it's using oxygen. Oxygen, why? It's the most electronegative element that we're dealing with. It really can draw those electrons far down from the state that they were in when they were in food. So lots of energy can be harvested. It's going to be the difference, the free energy available between the energy source and the terminal electron acceptor. Terminal electron acceptor, the final electron acceptor, the end of the electron transport chain. So we use oxygen. And so what happens when you don't get oxygen, those electrons don't have anywhere to go when they reach the end of the electron transport chain, and it all starts to back up. And when it backs up, you stop pumping protons. And when you stop pumping protons, you don't make ATP. And when you don't make ATP, you can't do cellular processes, and cells start to die. That's why you need oxygen. Now, not every living thing needs oxygen to do respiration. So you do have things called anaerobic respiration. Okay, they do anaerobic respiration by not using oxygen as their terminal electron acceptor. It's not as efficient, but it works. It's not as efficient because you don't have as big a differential between the free energy of the starting materials and the free energy of the final electron acceptor. So that's something to consider, but the principle is the same, and that's what I want you to think about. We happen to use oxygen. We have an oxygen-rich atmosphere. It's very, very electronegative. It's a great terminal electron acceptor, but it's not the only one. All right? So think about that and try to understand that concept with regard to oxidative phosphorylation.